Hi and Assalamualaikum. We are Group 2 and we will be presenting our lab proposal for Experiment 5, which focuses on coagulation and flocculation.
For background of study, surface water and wastewater will naturally contain dissolved and suspended particle, which leads to turbidity, which is a key indicator of poor water quality. So high turbidity is linked to diseases like diarrhea and skin cancer, and it will also affect the aquatic life by blocking the sunlight and reducing oxygen, making the water is unsuitable for domestic use. So in this experiment, the treatment that is being used is the coagulation and flocculation. So coagulation is a chemical process that destabilizes suspended particles using coagulants such as aluminium oxide, which allow the particles to cluster into a bigger flocks. The next one is flocculation, which is a gentle mixing process that will encourage the growth of flocks by promoting collusion and aggregation. So for problem statement, untreated water often contains tiny suspended particles and impurities that are too small for basic filtration to remove. So coagulation and flocculation help to group these small particles together so they can be easily removed. So in this experiment, coagulants play an important role in removing turbidity. The next one is objective. So the objective of this experiment is to examine the coagulation and flocculation method for treating wastewater. The next one is scope. The first one is to determine the effectiveness of coagulation and flocculation in removing turbidity, in reducing turbidity. The second one is to investigate the optimal amount of coagulant required to remove turbidity by varying the amount of coagulant into different beakers that contains the same water sample, and we will compare the turbidity and pH level of each beaker. For significance of experiment, so effective wastewater treatment will rely heavily on coagulation and flocculation, and this experiment will demonstrate that turbidity of the water can be reduced by adjusting the quantity of coagulant required. So this proved the critical impact of coagulant dosage on treatment efficiency of water sample.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Afin Aina binti Ahmad Razani, and I will continue this presentation for the methodology part. So the first one, we have the apparatus and materials that will be used in our experiment. So the first is the apparatus, is the main thing, jar test apparatus with the bird 6 pedal stirrer. And second one is the beakers. Also we have the graduated cylinders, pipettes, pH meter, turbidity meter, stopwatch, magnetic stirrer, thermometer, measuring spoons, glass rods for the manual stirring if needed, funnels or the filter paper, and also the safety equipment such as the gloves, goggles, and also the lab coats. So the materials is to use the raw water sample, UTM lake water, and also the civil water, civil faculty lake water, and then we have the coagulants, which are the aluminum sulfate and also the ferric chloride, and the flocculants is the polyelectrolytes, and the pH adjusting agents is the sodium hydroxide and also the sulfuric acid. And also we use distilled water for the solution preparation and the beaker cleaning. Next, we have the experimental procedures. So the first one, the plastic jar containing the wastewater sample will be shaken to ensure the uniformity, so that they don't have any substance in the bottom of it. And the second is the turbidity in NTU, and also the pH of the sample will be measured. 1 liter of the sample will be poured into 6 separate beakers. Label the beaker from 1 to 6. So the beaker 1 will be our control that will have none of the coagulant. So the coagulant will be used the aluminum sulfate. will be weighed and added to the remaining bakers in the following amounts according to the table 2.2. So as we can see here in the table, we have 6 beakers. So for the beaker 1, 0 coagulant. For 2, 3, 4, 5, 6, we have amount of the coagulant is amount of the coagulant are 0.2, 0.4, 0.6, 0.8, and 1.0 gram respectively. After the measure and pour into the beaker with the liquid water, and then we will put in at the jar test apparatus with the 6 pedals, and the pedals will be lowered into each beaker and set to 250 rpm. So once the speed has stabilized, the coagulant will be added simultaneously to each beaker, and then rapid mixing will be maintained for 1 to 3 minutes. And then the pedal speed will be reduced to 30 rpm and maintained for 30 minutes to facilitate the flow formation. After the slow mixing phase, the pedals will be removed and the mixture will be allowed to settle for additional 30 minutes. Okay. After that 30 minutes, the sample from each beaker will be taken using a syringe, and we will measure the turbidity and also the pH of each sample. For the safety procedures, for the experiments, we have three. So the first one is to wear gloves and goggles when handling aluminum sulfate and ferric chloride, as this is to avoid skin and eye irritation. And the second one is to add the coagulant carefully. This step is while the mixing. So we have to add it carefully to prevent the splashing during the mixing. And the third one is to dispose the treated wastewater and the chemical residues according to the proper laboratory waste disposal guidelines. And we also need to be very careful when do the cleaning of the beaker. So to avoid any beaker crack that may cause bleeding to our hands.
This is the analysis of the coagulation and flocculation experiment. The efficiency of water treatment will be analyzed using quantitative metrics derived from change in pH and turbidity before and after treatment. The following methods that calculation will be employ the mathematical equation pH efficiency, which is efficiency of pH in percent, initial pH minus with final pH times 100. And second one is turbidity removal efficiency. The efficiency of turbidity removal in percent, initial turbidity minus final turbidity times 100. And then this equation quantify the removal effectiveness of the treatment process. Turbidity is typically measured in NTU, means nephellometric turbidity units, and a reduction indicates successful particle aggregation and sedimentation. Next, we continue with the data analysis method. The experimental data will be collected at multiple stages of the treatment process. And then for each sample, we will measure the pH using the calibrated pH meter and turbidity using a nephelometer. These readings will be tabulated and the above equation will be applied to determine the removal efficiencies. The calculated efficiencies will allow comparison between different coagulant dosage and condition used in the experiment. And for the calculation, we need to use software and tools like Microsoft Excel or Google Sheet to organize data. So we perform calculation and generate graph from the Microsoft Excel.
Before conducting the experiment on coagulation and flocculation, we outlined a few anticipated results based on theoretical understanding. The main goal was to evaluate how effective the processes are in reducing turbidity and modifying pH levels in water sample. Let us start with turbidity removal. As we increase the dosage of a coagulant, we expect the turbidity to decrease significantly. This is because more suspended particles are bound together into flocks, which can then settle out. However, if too much coagulant is added, we may observe a drop in effectiveness. This is due to a phenomenon known as particle restabilization, where excess coagulant causes the particles to repel each other and remain suspended instead of settling. Next, regarding pH changes, since we are using acidic coagulant like alum, we expect the pH of the water to decrease during treatment. In most cases, the final pH will be lower than the initial pH, and depending on the buffer capacity of the water, this drop could be significant. This trend gives us a solid baseline for comparing our actual experimental results and evaluating the performance of the treatment process. Now, let us talk about how we plan to present the result from our coagulation and flocculation experiment. To make the data clear and easy to analyze, we'll be using both a table and a set of graphs. First, we will have a summary table labeled 5.2.1, which will show how different coagulant dosages affect key water quality parameters such as turbidity and pH. In addition to the table, we will include 4 key graphs to help visualize the data trends. This graph will clearly illustrate the relationship between anticipated result with the actual outcome and support any conclusion we draw later. Here we have table 5.2.1, which outline the relationship between the coagulant dosage, specifically aluminium oxide, and several water quality parameters. The first row represents the initial condition of the water sample before any coagulant was added. From there, each subsequent row corresponds to a different beaker with increasing amount of coagulant from 2 grams up to 10 grams. For each dosage level, we recorded the turbidity and pH of the treated water. We then calculated the changes in turbidity and pH compared to the initial value, labeled as delta turbidity and delta pH. To better understand the effectiveness of the process, we also calculated the percentage efficiency of turbidity removal and pH adjustment. This shows how well each dosage performed related to the untreated water. This table gives us a structured and detailed look at how different amounts of coagulant impact water clarity and acidity, which will be further analyzed in the graph and discussion that follow. As this is a proposal presentation, we have not conducted the experiment yet, but based on the literature review, we have outlined the graph we plan to generate once data collection is complete. So this graph will help us visualize the relationship between coagulant dosage and the changes in water quality parameters like turbidity and pH. So the first plane graph is turbidity versus coagulant. The second graph is pH versus coagulant. We expect a gradual decrease in pH as more coagulant is added, with the curve efficiency versus coagulant. This expected to rise sharply, reach an optimal and the level off or drop slightly if overdosing efficiency versus coagulation could increase with dosage, but is also expected to pH stabilize. This plan and they are grounded in established trends observed previously.